In the last video, I talked about SARS-CoV-2 virus, a coronavirus that causes the COVID-19 disease, and it's currently a pandemic, as you know. And I discussed that in terms of it being an evolutionary war, that is, the host trying to rid itself of the virus and the virus trying to get itself replicated and spread around. Today I want to talk about another pandemic. That was the 1918 Influenza A pandemic. Here are some of the Influenza A virions. On the right side you see an electron micrograph of the virus, kind of irregularly shaped because it has a host membrane around it, so it's somewhat fluid in its shape. It carries 8 chromosomes, RNA chromosomes, single-stranded. It also carries with it an RNA-dependent RNA polymerase to replicate its RNA. And then surrounding that are the matrix proteins that fill up the internal parts of the virus particle. Then there's the phospholipid bilayer from the host, and then a hemagglutinin trimer, and neuraminidase tetramer. Now, the hemagglutinin is a molecule that sticks to the host membrane, and that's how it gains access to the host cells. The various strains of influenza A circulate through natural hosts, mainly bird hosts, waterfowl and shorebirds. And it's pretty much endemic in those birds, and most of the time does not cause disease symptoms. They just carry it, and the chicks in particular have higher titers usually than the adults do. The virus is then easily transferred to domestic birds, including chickens and ducks and geese, etc. And it can also be passed on to mammals, including primarily swine, but also other mammals, and then eventually into humans. And then, of course, humans can transmit it to other humans, creating either a local epidemic or a pandemic. As I said, that the native hosts are waterfowl and other wild birds, especially ducks and geese and migratory waterfowl. There are 18 known strains or known types of the hemagglutinin gene and hemagglutinin protein, and 11 different types of the neuraminidase gene and neuraminidase proteins. Now, these can be transferred to various hosts, and you can see there the ones that are most prevalent in different species of organisms. Chickens are known to carry H5, 4, 7, 9, and 10. Humans usually have either H1N1, H2N2, or H3N2, although they've been known to be infected with H5N1, H9N2, and H7N7. However, the ones in red there, those indicate strains that can be picked up from the waterfowl but can't be transmitted from human to human. And in fact, those are the ones that most physicians and epidemiologists worry about, because if those ever change so that they can be transferred from human to human, those would be quite deadly and quite dangerous, probably similar to the current COVID-19 or even more severe. Here's a phylogram showing that there are 2 major groups of influenza strains, and the ones with the circles are the ones that humans primarily get. That's H1, H2, and H3. And then the ones that can be transferred from birds into humans are H5, H7, and H9 with the triangles. Most influenza affects the older population and the very young population primarily. But the 1918 influenza affected all groups, including people in their teens, 20s, 30s, and it was killing those people of all ages at similar rates. So it was quite a different type of influenza than had been seen previously in previous epidemics and pandemics. In that influenza pandemic, 500 million people were infected. Approximately a third of the world's population had been infected at that time, and it was probably even more than that. 50 million, I've seen some estimates up to 70 million people died around the world. 675,000 died in the U.S. alone. It was the only year that the population of the U.S. dropped. And 16 million died in World War I, just to show you the comparison. If the same thing happened today, approximately 250 million people would die. As a comparison, about 30 to 40, sometimes a little higher, sometimes a little lower, 30 to 40,000 people in the U.S. die from influenza every year. This was a particularly deadly influenza. Usually people died within 3 or 4 days, so they would become infected and sick on Sunday, and they'd be dead by Tuesday or Wednesday. As I talked about last time, this was caused by an extreme reaction by the immune system called a cytokine storm. So the people essentially suffocated to death, and you could see their lips turning blue and their hands turning blue as they lost oxygenation in their blood because their lungs were filling up with fluids. There are several theories about the origin of this pandemic influenza. It was an H1N1. One thing that was that they wondered at first was it a bird flu that then became able to transmit from one human to the other. And the sequences say not. It's just a human strain that became especially virulent. Was it the Spanish flu? No, the Spanish newspapers published a lot about this, but it was not in Spain. It did not originate in Spain. Did it originate in Fort Riley, Arkansas? Not likely. But did it arise in Asia? And that seems to be the most likely candidate. And then where did it go? It hasn't been seen since. So will it come back? Another question. So the most likely starting point was in China, and it has been reported in early 1917 that there was a respiratory disease that was spreading across northern China among farmers who usually kept chickens and pigs as part of the livestock for their trade. And by early 1917, it was killing about 200 people a day in northern China, which is a pretty high death rate. Central and northern China is along the route of many migratory birds, and so part of this strain could have come from wild birds, been transmitted into the chickens and then into the swine, and then you have a mixing of those different types to form a conglomeration of several different sequences from different sources. So in this case, it did have some human sequences from influenza from human sources, from swine sources, and from chicken sources, from avian sources. Influenza has 8 chromosomes, and each of the 8 chromosomes come together into the final particle, but the sources of each one of those chromosomes can come from a slightly different host. And so in China, in Asia, where they have pigs, chickens, and people, as well as other animals together, you can get, especially in the swine, the swine can be kind of a mixing bowl where it can contract sequences, viruses from the chickens, from other animals, from humans, and from other pigs. And so you can get what are called reassortants. That is, strains that have chromosomes from different hosts. This greatly increases the rate at which the influenza viruses can adapt to different species and create pandemic strains, thus increasing essentially the rate of evolution of those viruses. By late 1917, then, the disease was spreading to different parts of China and still killing about 200 people or more per day throughout China. Late in 1917 and in early 1918, Chinese workers were shipped by train and by boat into Europe and into Canada, and these were to help the Allies during World War I for the war effort. So they needed the soldiers to be on the front lines of the war, but behind the lines to do the cooking, supplying, and all the other types of work, they hired Chinese laborers to do that. As the Chinese laborers arrived in Europe and in Canada, many were ill and many were starting to die. In fact, a large proportion were dying by the time they arrived in Europe and Canada. And from there, the disease, this respiratory disease, which turned out to be influenza, started spreading. So by early 1918, many cities in the U.S. were affected, including military bases and forts, and that's why in Arkansas, that's why they thought maybe it started there. It also began spreading throughout Europe, so especially in France and Germany and other places during World War I. Many people were getting the disease, and a large proportion were dying. By mid-1918, it had spread, but the peak had gone down. So it started to look like things were subsiding. People started getting together more and thinking, okay, the war is over finally, and the influenza pandemic is over. We're over the worst part. But actually the worst was yet to come. And by late 1918, the worst of the pandemic hit, where about 80% or 90% of the deaths occurred during that time. Especially October was an extremely bad month for the number of deaths that occurred during that time. So here's a graph of how those peaks and valleys occurred. So you have the starting in 1918 in the spring, one peak, and then during the summer it went down, and then during the fall it started to come way up, including the peak here, which is in October. And then by the beginning of 1919 it began to decrease, and then in the summer, the number of deaths was quite low, and then it reappeared in the fall of 1919, and a fairly high peak, but not as high as the one in late 1918 occurred. Then by 1920, it was completely gone, and it has not come back since then. No one's seen that particular strain. There have been other H1N1 strains, but not that particular one. So, where did it go? So, the questions that have been asked then are, why did so many people become infected? And then why did so many die? And where did this virus come from? Where did it go? And can it return? So why did so many people become infected? Well, there was a very large susceptible host population. No one was immune to this particular disease. So the virus pretty much had free reign throughout the world. The way that influenza infects a host cell is that the hemagglutinin protein attaches to sialic acid residues on the host membrane, The host membrane then forms an endosome, taking the virus into that cell, and then the M2 ion channel acidifies the endosome, and that releases the viral genome as well as the viral proteins that are inside the virion. The virus polymerase makes new copies of the RNA as well as making the mRNA strands, which are then translated on ribosomes in the cytosol. The virus then starts to self-assemble on the host membrane and then buds off. You'll notice also the sialic acid residues on the right-hand side have been clipped off, and that's done by the virus so that the newly emerging virus particles don't attach to the same cells, but they'll only attach to new cells that they haven't infected previously. The hemagglutinin forms, as well as the sialic acid on the host membrane, come in different forms, so you can have different interactions there so that the cells become more or less receptive to the virion, the virus particle. The second question is, why did so many die? Why was this particular influenza so virulent and deadly to so many groups of people? As with the current SARS-CoV-2, some of the proteins that the virus produces cause an extreme immune response called a cytokine storm. This causes inflammation, blood vessels tend to dilate, fever can be caused, and also accumulation of fluids. And this one caused excessive fluids in the lungs, causing rapid pneumonias that would essentially suffocate the patients. And that's similar to what's happening with the COVID-19 disease currently. The third question is, where did it come from? And I've already discussed it probably originated somewhere in Asia. However, the mechanism by which it came about was probably via recombination, because there is frequent recombination, especially in these RNA viruses. There's also a lot of mutation, and also there's chromosome reassortment, which causes these reassortant varieties. And I've already discussed that, where you can get some chromosomes coming from an avian host, some coming from a swine host or other mammal, and then some coming from human hosts as well, creating completely new, essentially new types of virions, new strains. So just a little explanation about the genome. As I said, there are 8 chromosomes. Here they are. So the longest chromosome, PB, encodes a protein PB2 for transcriptase and cap binding. The second RNA fragment or chromosome also is part of the transcriptase, as well as a little part down there is for cell death, inhibits mitochondria. PA is a transcriptase and protease. HA is the hemagglutinin. NP is a nucleoprotein, which coats the RNA. NA is a neuraminidase, and then there are 2 other small fragments. One that produces the matrix protein, and the other one by splicing of the RNA makes the ion channel. And then the NS1 and NS2 are non-structural proteins involved in RNA transport, splicing, translation, and export of the viral RNA from the nucleus. So in addition to having some of these genes recombine coming from different hosts, you also can have reassortment of these separate fragments. So you can have the first 2 fragments coming from a human host, the next 4 coming from a swine, and the last 2 coming from an avian host, joining in one particle that then can probably infect humans, as well as avians and possibly swine as well. And this shows one of those events. This was the sort of construction of a reassortant that made the 2009 swine flu that caused a pandemic around the world and actually had a high death rate as well. So you can see that there was some mixing of swine flu, swine influenza here, and then some of human origin and some of avian or bird origin. Those mixed in about 2000 to form a couple of new reassortants, which contain combinations of fragments or chromosomes of swine, human, and avian origin. And then in 2009, or just slightly before that, you have more mixing within a swine host, adding other fragments or chromosomes from swine, producing the 2009 human H1N1 that caused a pandemic, which caused high morbidity and mortality. I happen to have gotten this one, and I can tell you that it was a very bad influenza. I had a temperature of 104 for 2 days. It then went down to 102 for 2 days, and I thought that I was not going to make it. It was really an unusual and severe flu, and then I coughed essentially for several weeks afterwards. And this was one of those that caused mortality in many different age groups, including 20 and 30-year-olds. The next question is, where did it go? And the answer there was no vaccine. So this had to do with herd immunity, getting enough of the human population to have antibodies to this, and natural selection. Several millions of people died because they didn't have immune systems that could handle this one. So in general, it disappeared because eventually enough of the human population became immune. It could not infect anyone after that, and it essentially went extinct. We haven't seen it since then. However, there have been other H1N1 varieties or strains, and there are currently H1N1 strains circulating now. And so the vaccine that people get nowadays consists of several different antibodies. One is for the H1N1, and then there's an H3 and another, I think, H1 type that is in the trivalent vaccine. So as I stated before, by 1920, at least 33% of the world population had gotten sick, and it was probably more than double that, actually. And 10% of those died. It was the only time in modern history where the U.S. population declined. And immunity and natural selection stopped the further spread of this and probably caused it to go extinct, at least that strain. Can it return? Well, according to some of our research, the answer is yes. One way is for it to sort of evolve again as a virulent strain, but we've also found that these viruses survive in ice for very, very long periods of time. So there is a biotic cycle that occurs in these migratory animals, birds and mammals as well. So they move north in the summer, in the spring and summer, to have their young, and they pick up viruses while they're in there, and then they go south for the winter into their wintering grounds and spread those viruses around, only to return the next spring carrying a new set of viruses with them. But we've also found there's an abiotic cycle which affects the evolution of these viruses. So the ice melts in the spring and summer, releasing viruses that had been deposited the year before. And then those viruses then freeze again. With the new viruses that are brought freeze. Those are held over the winter and then released again the next summer. And usually the birds that are leaving late in the season are not the same types of birds that arrive early in the spring and summer. So you get a mixing of the genotypes of these viruses throughout the different seasons. To demonstrate that, we obtained some ice samples, lake ice samples from Siberia, from eastern Siberia, from 3 lakes that are known to have bird populations, migratory bird populations. 3 lakes: Lake Adoma, Lake Sichichi, and Lake Park. Adoma and Sichichi have fewer birds because they're close to human habitation. Lake Park is very isolated. There are a lot of birds, and there are a lot of new chicks born every year. These lakes occur on major flyways, including those that connect East Asia, Central Asia, and the Pacific as well. And so it's a place where probably a lot of viruses are exchanged with these bird populations moving back and forth. So we sequenced some of the viruses from these lakes, and those are the ones listed in blue. All of these are H1 types, H1 influenza RNAs in the ice. All of the other ones are sequences from other studies that have been throughout the year. The 1918 influenza is up here, and you can see it's separated from all of the others. So it's good that it seems like that's one that may or may not come back. If you just focus in on the sequences of the influenza from Lake Park and Lake Adoma, on the phylogram they come out closest to influenzas from 1938, from 1933, and from 1967 and 39. So we think that these may have been deposited and they're still circulating in the birds, but they may have been frozen in the ice, and then they've come back at a later time to enter the bird populations from the melted ice. So more generally, we've come up with this idea of genome and organism recycling, especially with microbes, where they're deposited on glaciers or in any kind of environmental ice, lake ice, etc. Then they melt out at a later time. They're picked up by birds or by evaporation, wind, etc., and enter the current population of those species, enter hosts that they can infect, and then again repeat the cycle by being deposited again on the ice and in the snow. So they can essentially hide out for hundreds, thousands, tens of thousands, and even hundreds of thousands of years, and then reemerge at a much later time. And we've shown this in a couple different studies already, that this probably is occurring and has occurred. And now with global climate change and heating and melting of ice, a lot of organisms are currently being released. And this is another type of evolutionary process where you get recycling or reemergence of old genomes that mix into the population of the same organism with more modern or recent genomes. Okay, so that's the end of this little video on influenza. Hope you enjoyed it, and I'll talk to you later.